Hello everyone, let us start the chapter 8, Microbes in Human Welfare. In the previous videos, we have learned about human health and disease. Now we are talking about microbes in human welfare. Besides macroscopic plants and animals, microbes are the major components of the biological systems of the earth. You have studied about the diversity of living organisms in class 11. Do you remember which kingdom among the following living organisms contain the microorganism? Yes, microorganisms are contained in the Monera kingdom. We are talking about microorganisms. They can be viruses, bacteria, fungi. So they can be contained in protista, fungi, which are the ones that are only microscopic. Microbes are present everywhere in the soil, water, air, inside our bodies and that of our animals and plants. They are present even at the sites where no other life form could possibly exist. Sites such as deep inside the geysers, thermal vents where the temperature may be as high as 100 degrees Celsius, deep in the soil under the layers of snow several meters thick, and in the highly acidic environments like thermoacidophiles. High temperatures like thermophiles. Microbes are diverse: protozoa, bacteria, fungi, and microscopic animal and plant viruses. Viroids and also prions that are proteinaceous infectious agents. Prions are the proteinaceous infectious agents and viruses are the having the low molecular weight RNA, and these cause the potato spindle tuber diseases. Prions are the abnormally folded proteins. These are infectious agents and cause the mad cow disease and CR-Jacob disease in the humans. Microbes like bacteria and many fungi can be grown on nutritive medium to form colonies that can be seen with the naked eyes. Such cultures are useful in studies on microorganisms. Let us see some example. Bacteria, a rod-shaped bacteria magnified 1500 times. This is a rod-shaped bacteria, you can see, and these are magnified 1500 times. These are rod in shape. So second is spherical shaped magnified 1500 times. B part shows the spherical shaped bacteria magnified 1500 times. These are spherical shape. Third one is rod shaped bacteria showing flagella magnified 50,000 times. This is one rod shaped bacteria shown and showing the flagella also. So rod shaped bacterium is shown here. Now moving on to next figure, viruses. This is showing the viruses. First one is the bacteriophage. What are bacteriophages? Bacteriophages are the viruses infecting the bacteria. So this is the bacteriophage. Bacteriophage के parts आ सकते हैं, आपको फिगर आ सकता है उसमें लेबलिंग करने के लिए. So this one is the head. This is the collar. This is the tail. This is the plate. These are the pins. These are the prongs. जो पैर जैसे दिख रहे हैं, these are the prongs. टेल किसे बोलते हैं? जो एक पॉइंटेड स्ट्रक्चर दिख रहा है, दैट इज टेल. टेल के एंड पे क्या लगी है? प्लेट लगी है. प्लेट्स हैव द पिंस एंड कॉलर इज बिटवीन द हेड एंड द टेल. हेड एंड टेल के बीच में नेक होता है ना हमारा, देन इट इज़ अ कॉलर हियर. सो लेट्स सी नेक्स्ट इमेज. So B part is showing the adenoviruses which cause respiratory infections. So these are the adenoviruses causing the respiratory infections in organisms. Third one is rod-shaped tobacco mosaic virus, magnified about 1,50,000 times. These are the compact rod-shaped viruses. As you can see, these are rod-shaped. Let's see colonies of bacteria growing in a petri dish. These are the colonies of bacteria growing in a petri dish, and this is the fungal colony growing in a petri dish. So you can differentiate between the colonies of bacteria and the fungal colony. Fungal colony is confined to a small part only, and colonies of bacteria spread over the entire petri dish because bacteria are small and they can get diverged in the different regions. In chapter 7, you have read the microbes cause a large number of diseases in human beings. They also cause diseases in animals and plants, but this should not make you think that microbes are harmful. All microbes are harmful. Several microbes are useful to man in diverse ways. Some of the most important contributions of microbes to human welfare are discussed in this chapter. Microbes in household products. We have talked in biotechnology also. Some of the microbes like Bacillus thuringiensis and the Taq polymerase obtained from Thermus aquaticus. These are also used in the human welfare only, in the recombinant DNA and the biotechnology. Microbes in household products. You would be surprised to know that we use microbes or products derived from them every day. A common example is the production of the curd from the milk, the lab bacteria. Microorganisms such as lactobacillus and other commonly called lactic acid bacteria grow in the milk and convert it to curd. During growth, the lab produces acids that coagulate and partially digest the milk protein. A small amount of curd added to the fresh milk as inoculum or starter contains millions of larvae, which at suitable temperatures multiply, thus converting milk to curd, which also improves its nutritional quality by increasing the vitamin B12. In our stomach too, the lab play a very beneficial role in checking disease-causing microbes. The dough, which is used for making foods such as dosa and idli, is also fermented by bacteria. The puffed up appearance of the dough is due to the production of CO2 gas. Can you tell which metabolic pathway is taking place resulting in the formation of CO2? That is the anaerobic respiration. Where do you think the bacteria for these fermentation come from? These come from nature only. Similarly, the dough which is used for making bread is fermented using the baker's yeast Saccharomyces cerevisiae. A number of traditional drinks and food are also made by fermentation by the microbes. Toddy, a traditional drink of some parts of the southern India, is made by fermenting sap from the palms. Microbes are also used to ferment fish, soybean, bamboo shoots to make foods. Cheese is one of the oldest food items in which microbes were used. Different varieties of cheese are known by their characteristic texture, flavor and taste, the specificity coming from the microbes used. For example, the large holes in the Swiss cheese are due to the production of large amount of CO2 by the bacterium named Propionibacterium shermani. The Roquefort cheese are ripened by growing a specific fungi on them, which gives them a particular flavor. So Roquefort cheese is ripened by fungi, and Swiss cheese is ripened by a bacterium which produces a large amount of CO2, and the bacterium name is Propionibacterium shermani. Microbes in industrial product. Even in the industry, microbes are used to synthesize a number of products valuable to human beings. Beverages and antibiotics are some common examples. Production on an industrial scale requires growing microbes in a very large vessels called fermenters. Figure 8.8 shows the figure of the fermenters. These are the large vessels in which microbes are grown. This is the fermentation plant. Fermented beverages. Microbes, especially yeast, Saccharomyces cerevisiae, have been used from time immemorial for the production of beverages like wine, beer, whiskey, brandy or rum. For this purpose, the same yeast, Saccharomyces cerevisiae, or used for the bread making and commonly called the brewer's yeast, is used for fermenting malted cereals and fruit juices to produce ethanol. Malted cereals use होते हैं, fruit juices होते हैं to produce the ethanol. If you recollect the metabolic reaction which results in the production of ethanol by yeast, this was the fermentation reaction in the anaerobic respiration. Depending on the type of the raw material used for fermentation and the type of processing with or without distillation, different types of alcoholic drinks are obtained. Wine and beer are produced without distillation, whereas whiskey, brandy and rum are produced by distillation of the fermented broth. Moving on to antibiotics. Antibiotics produced by microbes are regarded as one of the most significant discoveries of the 20th century and have greatly contributed towards the welfare of the human society. Antibiotics are the most significant discoveries of the 20th century. Remember, 20th century में antibiotics were discovered किया गया था. Anti is a Greek word that means against, and bio means life. Together they mean against life in the context of the disease causing organism. So disease causing organism के context में देखिए तो antibiotic means against their life. because they are working against the life of the disease-causing microorganism. Whereas with reference to human beings, they are pro-life and not against. Antibiotics are chemical substances which are produced by some microbes and can kill or retard the growth of other disease-causing microbes. So you are familiar with the commonly used antibiotic, that is penicillin. Do you know that the penicillin was the first antibiotic to be discovered, and it was a chance discovery? Alexander Fleming, while working with Staphylococci bacteria, once observed the mold growing in one of its unwashed culture plates around which Staphylococci could not grow. So Staphylococci is basically a bacteria which was not able to grow around the mold growing on his unwashed culture plate. So he found out that it was due to the chemical produced by the mold, and he named it as penicillin after the mold Penicillium notatum. However, its full potential as an effective antibiotic was established much later by Ernest Chain and Howard Florey. The antibiotic was extensively used to treat American soldiers wounded in World War II. Fleming, Chain and Florey were awarded the Nobel Prize in 1945 for this discovery. After penicillin, other antibiotics were also purified from other microbes. Can you name some of the antibiotics? Find out their sources. Antibiotics have greatly improved our capacity to treat deadly diseases such as plague, whooping cough. It is also known as Kali Khasi because cough is Khasi and this is Khasi. So diphtheria is known as Galgotu and leprosy, that is known as Kushcherog, which used to kill millions all over the globe. Today we cannot imagine a world without antibiotics. We get a small disease or we already take the antibiotic nowadays. So chemicals, enzymes and other bioactive molecules. Microbes are also used for commercial and industrial production of certain chemicals like organic acids, alcohol and enzymes. Example of acid producers are Aspergillus niger, a fungus of citric acid, Acetobacter acidi, a bacterium of acetic acid, Clostridium botulinum, a bacterium of butyric acid, and Lactobacillus, a bacterium of lactic acid. Yeast, Saccharomyces cerevisiae, is used for commercial production of ethanol. Microbes are also used for the production of enzymes. Lipases are used in detergent formulations and are helpful in removing oily stains from the laundry. You must have noticed that the bottled juices bought from the market are clearer as compared to made at the home. This is because the bottled juices are clarified by the use of pectinases and proteases. Streptokinase is produced by the bacterium Streptococcus and modified by genetic engineering is used as a clot buster for removing the clots from blood vessels of patients who have undergone myocardial infarction leading to the heart attack. Another bioactive molecule, cyclosporin A, which we have talked about, immunosuppressive agent for the graft rejection, cell-mediated immunity. So that is used as an immunosuppressive agent for the organ transplant patients. Is produced by the fungus Trichoderma polysporum. Statins produced by the yeast Monascus purpureus have been commercialized as blood cholesterol lowering agent. It acts by competitively inhibiting the enzyme responsible for synthesis of cholesterol. So statins are produced by yeast Monascus purpureus, whereas cyclosporin is produced by a fungus Trichoderma polysporum. Streptococcus bacteria produces streptokinase enzyme for myocardial infarction, and pectinases and proteases are produced for clearing the bottled juices. In detergents or laundry, to remove the oily stains, we use lipases. And yeast Saccharomyces cerevisiae is used in the commercial production of ethanol. It is also used in the bakers. It is also known as the baker's yeast or the brewer's yeast. So Aspergillus niger, a fungus of citric acid, Acetobacter acidi, a bacterium of acetic acid, Clostridium botulinum, a bacterium of butyric acid, and Lactobacillus, a bacterium of lactic acid. Microbes in waste treatment. We know that large quantities of wastewater are generated every day in cities and towns. A major component of this wastewater is the human excreta. This municipal wastewater is also called sewage. It contains large amounts of organic matter and microbes, many of which are pathogenic. Have you ever wondered where this huge quantity of sewage or urban wastewater is disposed of daily? No. This cannot be discharged into the natural water bodies like rivers and streams directly. You can understand why, because it may contaminate the water, or वही drinking water we are drinking, so उसकी वजह से it will spread many infectious diseases through water only. Before disposal, hence sewage is treated in sewage treatment plants to make it less polluted. Treatment of wastewater is done by the heterotrophic microbes naturally present in the sewage. So this treatment is carried out in 2 stages. So this is the figure showing the secondary treatment. Question may be asked, the figure denotes the primary treatment, secondary treatment, sewage treatment, or the tertiary treatment. So answer will be secondary treatment. So let's talk about the stages of treatment. These are 2 stages: primary treatment and secondary treatment. Primary treatment, these treatment steps basically involve physical removal of particles large and small from the sewage through filtration and sedimentation. We will do filtration and sedimentation. These are removed in stages. Initially, floating debris is removed by sequential filtration. Then the grit, soil and small pebbles are removed by sedimentation. All solids that settle from the primary sludge and the supernatant forms the effluent. So the effluent from the primary settling tank is taken for the secondary treatment. Secondary treatment or biological treatment, the primary effluent is passed into large aeration tanks where it is constantly agitated mechanically and air is pumped into it. This allows vigorous growth for useful microbes, aerobic microbes, into flocks, masses of the bacteria associated with the fungal filaments to form mesh-like structures. While growing, these microbes consume the major part of the organic matter in the effluent. This significantly reduces the BOD, biochemical oxygen demand of the effluent. BOD refers to the amount of oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by the bacteria. Remember the definition of BOD. BOD का full form भी याद रखना, biochemical oxygen demand. This is given in the NCERT. Sometimes it is written biological oxygen demand. So it is incorrect as per the NCERT. So BOD refers to the amount of oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by the bacteria. The sewage water is treated till the BOD is reduced. The BOD test measures the rate of uptake of oxygen by microorganism in a sample of water, and thus indirectly BOD is the measure of organic matter present in the water. The greater the BOD of wastewater, more is its polluting potential. So secondary treatment में क्या हो रहा है? Secondary treatment is also known as biological treatment. इसमें क्या होता है, जो हमारा primary effluent था that is passed into large aeration tanks, जहाँ पे वो constantly agitate किया जाएगा mechanically और air pump की जाएगी उसमें. उसकी वजह से vigorous growth हो जाएगी aerobic microbes की, because air pump कर रहे हैं, oxygen pump कर रहे हैं. So aerobic microbes की vigorous growth हो जाएगी into the flocks. What are flocks? Flocks are the masses of bacteria associated with the fungal filaments to form mesh-like structures. So they will consume the major part of the organic matter in the effluent. इसकी वजह से BOD जो है डिक्रीज हो जाएगी because organic matter will be decreasing. So BOD refers to the amount of oxygen that will be consumed if all the organic matter in 1 liter of solution was oxidized by bacteria. So sewage water तब तक treat करेंगे जब तक BOD reduce ना हो जाए. Once the BOD of the sewage or wastewater is reduced significantly, the effluent is then passed into the settling tank where the bacterial flocks are allowed to sediment. This sediment is called the activated sludge. A small part of the activated sludge is pumped back into the aeration tank to serve as the inoculum. Inoculum मतलब एक small part जो है हम उसे ऐड कर देंगे aeration tanks में, जिसकी वजह से easier growth हो जाएगी बैक्टीरिया की. Easier growth हो जाएगी fungi and bacteria mesh-like structures called the flocks. The remaining major part of the sludge is pumped into large tanks for anaerobic sludge digester. Here, other kinds of bacteria which grow anaerobically digest the bacteria and the fungi into the sludge. During this digestion, bacteria produce mixture of gases such as methane, hydrogen sulfide, and carbon dioxide. These gases form biogas and can be used as a source of energy as it is inflammable. The effluent from the secondary treatment plant is generally released into the natural water bodies like the rivers and the streams. And aerial view of such plant is shown in the figure 8.7. This is an aerial view of sewage treatment plant. So you can appreciate how microbes play a major role in treating millions of gallons of wastewater every day across the globe. Millions of gallons of wastewater. Okay, this methodology has been practiced for more than a century now in almost all parts of the world. Till date, no man-made technology has been able to rival the microbial treatment of sewage. You are aware that due to the increasing urbanization, sewage is being produced in much larger quantities than even before. However, the number of sewage treatment plants has not increased enough to treat such large quantities, so the untreated sewage is often discharged directly into the rivers, leading to their pollution and increase in the waterborne diseases. Waterborne diseases like ascariasis. The Ministry of Environment and Forest has initiated Ganga Action Plan and Yamuna Action Plan to save these major rivers of our country from pollution. Under these plans, it is proposed to build a large number of sewage treatment plants so that only the treated sewage may be discharged in the rivers. A visit to the sewage treatment plant situated in any place near would be very interesting and educating experience. So microbes in the production of biogas. Now let's talk about the biogas production using the microbes. Biogas is a mixture of gases containing predominantly methane, produced by the microbial activity and which may be used as a fuel. You have learned about the microbes produce different types of gases and products during growth and metabolism. The types of the gas produced depends upon the microbes and the organic substance they utilize. In the example cited in the relation to fermentation of dough, cheese making and production of beverages, the main gas produced was carbon dioxide. However, certain bacteria which grow anaerobically on cellulosic material may produce large amount of methane along with CO2 and H2. So, certain bacteria which grow anaerobically on cellulosic material produce large amount of methane and carbon dioxide and hydrogen. These bacteria are collectively called methanogens, and one such common bacterium is the methanobacterium. These bacteria are commonly found in the anaerobic sludge during the sewage treatment. These bacteria are also present in the rumen, a part of the stomach of cattle. A lot of cellulosic material present in the food of the cattle is also present in the rumen. In rumen, these bacteria help in the breakdown of cellulose and play an important role in the nutrition of the cattle. We humans are able to digest the cellulose present in our foods because of the probiotics present in our stomach. Thus, the excreta dung of the cattle, commonly called the gobar, is rich in these bacteria. Dung can be used for generation of biogas, commonly called gobar gas. The biogas plant consists of a concrete tank 10 to 15 feet deep, in which bio-waste are collected and slurry of the dung is fed. A floating cover is placed over the slurry, which keeps on rising as the gas is produced in the tank due to the microbial activity. The biogas plant has an outlet which is connected to a pipe to supply biogas to nearby houses. The spent slurry is removed through the another outlet and may be used as a fertilizer. Cattle dung is available in large quantities in rural areas where cattle are used for a variety of purposes. So biogas plants are more often built in the rural areas. The biogas thus produced is used for cooking and lighting. The picture of the biogas plant is shown in the figure. The technology of biogas production was developed in India mainly due to the efforts of Indian Agriculture Research Institute and Khadi and Village Industries Commission (KVIC) and IERI. If your school is located near a village, you will see the biogas plants. So this is a typical biogas plant. It is a digester. Dung and water. This is the place where slurry is fed. Slurry comes in the concrete tank, which is 10 to 15 feet deeper, and it consists of digester. Here the anaerobic respiration takes place, and the sludge is taken out from here. This is the gas holder having the cover. So the cover keeps on rising as the gas is produced in the tank due to the microbial activity, and gas is finally supplied to the household through the pipeline. So the biogas mainly consists of biogas is a mixture of gases, predominantly methane. It also contains carbon dioxide and hydrogen. Moving on to microbes and biocontrol agent. Biocontrol refers to the use of biological methods for controlling plant diseases and pests. In modern society, these problems have been tackled increasingly by the use of chemicals, by use of insecticides and pesticides. These chemicals are toxic and extremely harmful to human beings and animals alike, and have been polluting our environment, soil, ground, water, fruits, vegetables and crop plants. Our soil is also polluted through use of the weedicides to remove the weeds. So biological control of pests and diseases. In agriculture, there is a method of controlling pests that relies on the natural predation rather than the introduced chemicals. A key belief of the organic farmer is that the biodiversity furthers health. Key belief of the organic farmer is that the biodiversity furthers health. The more variety a landscape has, the more sustainable it is. The organic farmer therefore works to create a system where the insects that are sometimes called the pests are not eradicated, but instead are kept at the manageable levels by a complex system of checks and balances within a living and the vibrant ecosystem. Contrary to the conventional farming practices, which often use chemical methods to kill both useful and harmful life forms indiscriminately, this is a holistic approach that seeks to develop an understanding of the webs of interaction between the myriads of organisms that constitute the field fauna and the flora. The organic farmer holds the views that the eradication of the creatures that are often described as pests is not only possible but is also undesirable, for without them the beneficial predatory and the parasitic insects which depend upon them as the food or host would not be able to survive. Thus, the use of biocontrol measures will greatly reduce our dependence on toxic chemicals and pesticides. An important part of the biological farming approach is to become familiar with the various life forms that inhabit the field, predators as well as the pests, and also their life cycles, the patterns of feeding and the habitats that they prefer. This will also help develop appropriate means of biocontrol. The very family of beetle with red and black markings, the ladybird, and the dragonflies are used to get rid of the aphids. Ladybirds are used to get rid of aphids, and dragonflies used to get rid of mosquitoes respectively. An example of microbial biocontrol agents that can be introduced in order to control butterfly caterpillars is the bacteria Bacillus thuringiensis, often written as BT. These are available in sachets as dried spores which are mixed with water and sprayed onto the vulnerable plants such as brassicas and fruit trees. So remember, vulnerable plants जो होते हैं इस केस में these are brassicas and fruit trees, where these are eaten by the insect larvae. In the gut of the larva, the toxin is released and the larva get killed. The bacterial disease will kill the caterpillars but leave other insects unharmed. Because of the development of methods of genetic engineering in the last decade or so, the scientists have introduced Bacillus thuringiensis genes into the plants. Such plants are resistant to the attack by the insect pest. So, BT cotton is one such example, which is being cultivated in some states of our country. A biological control being developed for use in the treatment of plant disease is the fungus Trichoderma. Trichoderma species are free-living fungi that are very common in the root ecosystem. They are effective biocontrol agents of several plant pathogens. Baculoviruses are pathogens that attack the insects and the other arthropods. The majority of baculovirus used are as biocontrol agents and are in the genus Nucleopolyhedroviruses. These viruses are excellent candidates for species-specific narrow-spectrum insecticidal applications. Species-specific narrow-spectrum insecticidal applications, they have been shown to have no negative impacts on plants, mammals, birds, fish, or even on non-target insects. This is especially desirable when beneficial insects are being conserved to aid in the overall integrated pest management program, or when an ecologically sensitive area is being taken. Microbes as biofertilizers. With our present-day lifestyles, environment pollution is a major cause of concern. The use of chemical fertilizers to meet the ever-increasing demand of the agricultural produce has contributed significantly to the pollution. Of course, we have not now realized that there are problems associated with the overuse of the chemical fertilizers, and there is large pressure to switch to organic farming, the use of biofertilizers. Biofertilizers are organisms that enrich the nutrient quality of the soil. The main sources of biofertilizers are bacteria, fungi, and cyanobacteria. You have studied about the nodules on the roots of leguminous plants formed by the symbiotic association of rhizobium. These bacteria fix atmospheric nitrogen into the organic form, which is used by the plant as a nutrient. Other bacteria can fix atmospheric nitrogen while free living in the soil. Example, azospirillum or azotobacter, thus enriching the nitrogen content of the soil. So, nitrogen fixation is done by azospirillum and azotobacter, and these are free living in the soil. Symbiotic association is formed by rhizobium, and the main sources of biofertilizers are bacteria, fungi, and cyanobacteria. Moving ahead, fungi are also known to form symbiotic association with the plant's mycorrhiza. Many members of the genus Glomus form mycorrhiza. The fungal symbiont is in these associations absorb phosphorus from the soil and passes it to the plant. Plants having such associations show other benefits also, such as resistance to the root-borne pathogens, tolerance to salinity and growth, and an overall increase in the plant growth and development. Can you tell what advantage the fungus derives from the association? The fungus get the shelter and the food from the plants. Cyanobacteria are autotrophic microbes widely distributed in aquatic and terrestrial environments, many of which can fix atmospheric nitrogen. Example, Anabina and nostoc and oscillatoria. In paddy fields, cyanobacteria serve as an important biofertilizer. Blue-green algae also add organic matter in the soil and increase its fertility. Currently in our country, a number of biofertilizers are available commercially in the market, and farmers use these regularly in the fields to replenish the soil nutrients and to reduce the dependence on the chemical fertilizers. Let's talk about summary. Microbes are a very important component of life on earth. Not all the microbes are pathogenic. Many microbes are very useful to the human beings. We use microbes and microbially derived products almost every day. Bacteria called the lactic acid bacteria grow in the milk to convert the milk into the curd. The dough which is used to make bread is fermented by the yeast Saccharomyces cerevisiae. Certain dishes such as idli and dosa are made from the dough fermented by microbes. Bacteria and fungi are used to impart particular texture, flavors and taste to the cheese. Microbes are used to produce industrial products like lactic acid, acetic acid, alcohol, which are used in variety of processes in the industry. Antibiotics like penicillin produced by useful microbes are used to kill disease-causing harmful microbes. Antibiotics have played a major role in controlling the infectious diseases like diphtheria, whooping cough, and pneumonia. For more than 100 years, microbes are being used to treat sewage wastewater by the process of activated sludge formation and helping the recycling of water in nature. Methanogens produce methane while degrading plant waste. Biogas produced by the microbes is used as a source of energy in rural areas. Microbes can also be used to kill pests, the process known as the biocontrol. The biocontrol measures help us to avoid the heavy use of the toxic pesticides for controlling pests. There is the need these days to push for the use of biofertilizers in place of the chemical fertilizers. It is clear from the diverse uses human beings have put microbes so that they play an important role in the welfare of the human society. So this was all about the chapter Microbes in Human Welfare. I hope you all understand this chapter. So stay tuned on this channel for the more upcoming revision lectures. Thank you for listening me out. Have a great day.